Hello, my name is Marc López Cano, and I am a biomedical researcher at the University of Barcelona. Today, I would like to give you some hints about animal research. As you may know, over the last decades, medicine has experienced a great progress in many fields. The most recent one is the rapid production of vaccines against the COVID-19 based on RNA, which will hopefully allow all of us coming back to our pre-pandemic daily life. But it is not the only case in the field of vaccine development. Some other infectious disease that caused in the past many deaths has been almost eradicated, or their impact in human health has been significantly reduced. There are many other examples. Current treatments against many types of cancer are much more effective and safer for patients than classic ones. Clinicians have a wider range of drugs to prescribe for mental disorders, metabolic diseases, or pain relief. Surgery techniques, including organ transplantation or joint prosthesis implantation, have also evolved to offer better prognosis to many patients. All these advances have positively impacted in the health and quality of life of many people around the world. And it is important to know that all these progresses would have not been possible without animal research. Perhaps you may have some concerns about the use of animals for experimental purposes. Are animals still necessary in the 21st century? Does not exist other alternatives to replace animal-based experiments? And more important, how can investigators be sure that animals are not suffering in their experiments? If you want to follow me, we will have the chance to answer these questions by knowing better the work of some of my colleagues here at the University of Barcelona. So here we are with Dr. Francisco Ciruela from the Neuropharmacology and Pain Group. So good morning, Dr. Francisco.
Good morning.
Thank you for agreeing to make us aware of your research. And please, could you tell us which are your main research goals?
Okay, so the research we do in the laboratory is devoted to the design of new drugs which will be safer, more effective for the treatment of human diseases, and eventually also to take care of the animals we work with.
Thank you. So, and why is it necessary for your research to use animals?
We need to use animals as we need to model human diseases to study the effects of drugs into these diseases. So we work mostly with diseases that affect the central nervous system, including Alzheimer's disease, Parkinson, and motor neuron diseases. And this is the only way we can model those diseases into animals to study the drug effects on those.
So according to that, for you it's completely mandatory to use animals. Which measures do you implement to ensure the animal welfare?
So we are subjected to the European laws regarding the animal research. Those laws are driven by the local committee, ethical committee of the University of Barcelona, and also supervised by the Catalan government ethical committee, which approve all protocols to for the management of the animals.
And which are the specific measures that you implement?
So the specific measures could be resumed into the 3R concept. The 3R concept is based on the reduce, refinement, and replacement of animals for research. And a specific interesting issue is in the refinement is also to introduce the use of animals for extracting more information. So this will produce a reduction in the number of animals we use in our research.
Now we are with Dr. Beatrice Terni from the Neurobiology Group. So thank you, Beatrice.
Welcome.
Thank you for agreeing to answer our questions. And Please, can you tell us which are your main goals in your research?
So in the lab we are interested in studying the lesion of the nervous system and the mechanism to repair them. So as you know, the traumatic brain injury as can occur after a car accident or any other traumatic event impact in the quality of life of people affected by them. And the reason is because the humans are not able to repair this sort of injuries. So on the contrary, there are some animals as Xenopus tadpoles that can do it very well. So in the lab we are using this animal model to investigate the mechanisms they use to repair this lesion and to recover quite quickly the functionality of the affected part with the goal of learning by them, so to know how they do it and how they do it quite well. And the aim of our investigation is to design in the future an experimental approach for therapeutical approaches for applicable to humans. So this is the main goal of our project.
So thank you very much for your clarification. So then for your group it's completely mandatory to use animal models,
yes,
right?
Yes. So the animal is the key point of our research. So without this animal we couldn't answer any question of our investigation, and because there are questions that cannot be addressed with an in vitro culture or a pure in vitro system. So we really need to see what happened in a living organism. This is the reason why we can do our research focus on the use of the animals, of this specific animal model.
So given that you cannot replace the animals in your experiments, what other measures do you implement to ensure the animal welfare?
Well, first of all the experimental procedures follow the European regulation for animal research. And then for the experiment we try to use the same animal for different type of experiment. For example, we can use one tadpole for behavioral studies, and then the same tadpole can be used, for example, for morphological analysis. And then what is important is the statistic analysis. So with statistics we try to minimize the number of animals needed for each experimental group.
So and now we are with Dr. Miguel Hueso from the Neurology Group. So thank you, good morning, and thank you for making us aware of your research. And could you explain us, please, which are the main goals of your research?
We are working in the inflammatory mechanisms of vascular lesions and arteriosclerosis who cause renal and heart diseases. Our main goal is to find some immunomodulatory target or molecule who could be used like a drug to try to change the normal history of the vascular lesions. We are also very interested in using alternative methods, so we are aware that there are organs on chip that in the future maybe could replace the use of animal models, but in the current situation we need animal models to test this hypothesis.
So knowing that it's important or mandatory for your group to use animal models, and you have talked about the replacement, but which other measures do you implement to ensure the complement of the 3R concept?
We try also to minimize the use of the animals in our experiments. As I told you before, we use cellular models before to jump to the animal model, and later we calculate the minimum number of animals that we have to use based in our expected results. Moreover, we have designed a multi-step strategy, so we are analyzing small groups of animals since we detect our expected results, so we don't need to use all the theoretical number of animals to test our hypothesis. We are very conscious of the importance of animals in our research, we take care of the animals as our patients. So we are trying to avoid the suffering, trying to avoid all the stress in this. We are very conscious that experimental in animals will have future treatments for our patients.
Thank you very much for your clarification. So now we are going to talk with one of my colleagues, Claudia Fernandez, from the Diabetes and Experimental Endocrinology Group. So thank you, Claudia, for your collaboration and for making us aware of your research. Could you please explain to us which are the main goals of your research?
Yes, thank you for inviting me. And our main goals of the research that we do here are directed to diabetes. So there's type 1 and type 2 diabetes, and the important thing here is to know that nowadays there are 500 million people that are suffering this pathology, and it's expected that in 2045 the number of people will increase to 700 million people. So it is really important to understand how this pathology works and the mechanisms to be able to cure diabetes. In this case, what we are trying to do here is implement the islet transplantation for type 1 diabetes, and what we use here is the animal research animals in order to mimic and to recapitulate what happens whenever we use this therapy to cure type 1 diabetes.
So, Claudia, taking into account that you cannot avoid the use of animals for your research, how does these animal models impact on your research?
So, animal research is essential in our studies and our research, but first we use cell lines in our case in order to mimic what happens in diabetes. So we put those cells in high glucose, for example, or we can use other types of drugs in order to understand what happens in those patients that suffer with diabetes. But in any case, at the end of the study, we need to implement that using animals, research animals, because it's the only way for us to replicate, in our case, the islet transplantation. We need to use in vivo models, and in this case, that's why we implement the animal research models.
So thank you, Claudia, for your clarification. And you have talked about the replacement, but which other measures do you apply in your research regarding, for example, the number of the animals?
Yes, so that's a good question. We always use the least amount of animals in our research in order to minimize the number of animals that are used. And in any case, the animals that we use, they follow strict rules under the ethical committee of this our research center. And what they always use is we always use is analgesia and anesthesia in order to maintain those animals in a good welfare. And also, it is important to know that the number of animals needs to be statistically significant in order to obtain the results that we want to obtain, and solid results in this case.
And finally, we are going to talk with Dr. Ricardo Perez from the Cell Biology Unit. So thank you, Dr. Ricardo Perez, for agreeing to share with us your knowledge. And please, could you explain to us which are the main goals of your research?
And it's a pleasure to meet with you and speak about our research and the implication of research in animal practice. So we are working here in the Cell Biology Unit, and our group is related with cancer cell biology goal. So the idea is to understand better the effect of different new drugs for cancer treatment, and the develop of new drug in collaboration with a chemistry group So mainly we are focusing in lung cancer, gliomas, and other like breast cancer. That is all.
So regarding your research, how does animal research impact your activities?
The animals and the research in animal is very important for our research area, because as I explained previously, we are working to analyze and discover or try to obtain new compounds from cancer treatment, mainly, as I told you, in lung cancer, gliomas, and breast cancer. Consequently, after the treatment with other approach like cell culture, organoids, etc., we need to move immediately to the animal unit to identify the effect of this new promised drug for cancer treatment. So we have different models in order to identify the effectivity of the drug, of the compound in this group of cancers.
Thank you very much.
You're welcome.
After listening to my colleagues working at different areas such as cancer, diabetes, neuroscience, and nephrology, I hope you will agree with me that animal research is an essential activity for improving health, and that scientific community is completely committed to reduce the animal suffering in their research experiments.